Hi IB students, we are grade 11 students from Beijing Huijia Private School, and we are studying biology courses in Mr. Diaram's class. Today we are going to talk about DNA replication, transcription, and translation. There are 3 people in our biology group. I am Peggy, this is Jack, and this is Jessica. I will introduce you the nature of semi-conservative DNA replication. There were 3 models suggested to explain the structure of double helix of DNA: conservative, semi-conservative, and dispersive. Conservative model suggests that the parental strands are together and the newly formed daughter strands are together. Theory of semi-conservative replication proposed that both of the parental strands work as the template for the new complementary strand, which means each DNA composed of an original strand and a new strand after replication. Dispersive model suggests that DNA strands are composed of double-stranded segments. Matthew Matheson and Franklin Stiles carried out an experiment that aimed to prove the model of semi-conservative. They cultured the bacteria E. coli for 14 generations in a medium containing a heavy isotope of nitrogen N15, which has one more neutron than N14 isotope. Then the E. coli was shifted in medium containing N14. The results supported semi-conservative replication model, as there is an increasing amount of DNA contains N14 only. Semi-conservative replication happens as the double helix of parental strands separate and new strands are made to match the separated strands. The separation is carried out by helices, the enzyme that breaks the hydrogen bonds between complementary bases. The 2 strands of DNA run antiparallel. Leading strand grows continuously from 5' to the 3', and lagging strand grows discontinuously from the 3' to the 5', and the new bases are added to the 3' end of the growing chain. Primates provide the starting point for DNA polymerase to begin synthesis of the new strand. For lagging strand, RNA primerase attaches to the DNA and synthesizes a short RNA primer with a free hydroxyl group. DNA polymerase links nucleotides together by making hydrogen bonds between complementary bases. DNA ligase rejoins two DNA strands into a double helix and joins Okazaki fragments of the lagging strand by forming a phosphodiester bond.
In this part, let's talk about DNA transcription. The purpose of transcription is to make mRNA, which is a messenger to carry information to make protein. DNA will help to synthesize mRNA copied from DNA sequence by RNA polymerase. So first, it must use a DNA section as a template to make mRNA. The enzyme RNA polymerase binds to the site of DNA section. This kind of enzyme move along DNA and separate them into single strands. And because RNA is single-stranded, so they only use one side of DNA as a template. The DNA strand that have the same base sequence with mRNA is called sense strand, and the other strand is called antisense strand. And then they'll pair up free RNA nucleotides to make mRNA. The mRNA will be complementary bases on the DNA strand. Because there's no silane in mRNA, so your cell will pair with adenine. After the RNA polymerase form covalent bond between those RNA nucleotides and reach the end of DNA section, the transcription will stop and a complex chain of RNA molecule will be released. RNA will separate from DNA and the double helix will reform.
Then I will explain DNA translation. There are 3 main parts about the translation, which is initiation, elongation, and termination. Those 2 gray stuff are the ribosomal subunits. The gray part of the mRNA is the start codon and the red one is the stop codon. During initiation, The components of the translational apparatus come together with an mRNA, and the tRNA carrying the first amino acid binds to the start codon. Anticodons are three opposite bases which correspond with codons on mRNA. During elongation, amino acids are brought to the mRNA by tRNAs and are added one by one to a growing polypeptide chain. During termination, a stop codon in the mRNA is recognized by a protein release factor. And the translational apparatus comes apart, releasing a complete polypeptide